Differential expression is probably the most important part of data analysis. And here we can do that in two different approaches, or addressing two different questions. The first one is to identify gene markers to distinguish cell populations. And the other one is to identify differentially regulated genes across conditions. In our case is COVID versus control. And we do so by using a function called rank genes group. And there we can set the test either parametric or non-parametric. And today we are going to test Wilcoxon as non-parametric and t-test as parametric. So first let's load the data and see what we have in our data sets. So we have 7,000 cells and 19,000 genes. And this is the resolution that or UMAP that we are using. And we are going to compare each cluster versus others to see whether there are given genes showing significant difference compared to the other clusters. We do that by t-test. Something important which I forgot to say is that please note that by default the raw data in AND-data object is used. So we can control that by specifying this parameter. Just for sanity check, let's see what we have in our AND-data. AND-data, let's print AND-data x and that. So these are normalized values because they have decimals. So when we are going to run rank gene test, we specify use raw to be false. There we go. We run the test and data, the clustering information, the method t-test, and key added to the output will be t-test. And then we plot it. It's the same the same function name. And then we say, okay, use the data, the t-test output or differential expression output. Visualize the top 25 genes for each clusters. And then the key will be t-test. So here we go. This is the top 25 genes and their scores. So they are the score, the more significant difference showed between the cluster 0 and the rest of the clusters. Cluster 1, the rest of the clusters, and so on. So we had 12 clusters from 0 to 11. And here we see these top 25 genes in each cluster. You can also extract them as a table, so using get rank genes groups as a data frame. So we have the data and then we specify which group. We are looking into cluster 1, for example. And the key is t-test. We set a p-value cutoff for 0.01 and then the log to full change as 0. So that's probably not the best settings, but just to show you as an example. So here we end up 3,423 rows and 5 columns. Another test that we can use is t-test overestimated the variance. So it performs t-test for differential expression, but overestimates the variance, which helps to make the test a bit more conservative and reduce false positives. We can run this, and then for visualization, let's copy paste this one, for example. And then we add this overestimate. I need underscore. There we go. So it should show the same. So if you look at this, for example, lies still pops up S100A9. Let's see in the same S100A9. Yeah, but it is a bit tighter scores or values when we compare it. So this is a bit more conservative approach if you want to use parametric tests. Now let's talk about Wilcoxon rank-sum. It is a non-parametric test. It is the same settings that we are using. And then here we should also set use raw as false. let's see if the same two genes that we just discussed shows up in the output. Slightly different order, but we have them. And as you can see, it's a bit more linear compared to the parametric test. So which one to choose? It's a difficult question, but we can try to address that by making a comparison between them. And we focus on genes differentially expressed in cluster 1. So we generated 3 data tables, data frames. The first one will be t-test, and here we extract the names. Pay attention that we are using exactly the same cutoff for log fold change and also the p-value cutoff. T-test over estimated variance, and then Wilcoxon. And then we generate a Venn diagram to see, okay, how much overlap exists between these 3 approaches. So to large extent, the large fraction of differential expressions have been captured with all of these 3 methods. Each of the tests have been able to identify unique differential expressions, but which one is the true one is very difficult to say. Usually the Wilcoxon test, which is a non-parametric test, works fine, and I usually most of the time use Wilcoxon unless there is a strong motivation to use the other tests. Now let's do some visualization. We are going to generate a heat map, a dot plot, a violin plot, and a matrix plot. And for each of them we use different functions, and there we specify to show top 5 genes, and also we specify to group them by the clustering we have in Leiden 0.6. And then we also specify for the heat map to show the gene labels. So the first one is the heat map. So we have the clusters, labeling of the clusters on the y-axis, and then the x-axis we have the genes. And we have the dendrogram showing the order or closeness or correlation of the clusters in relation to the expression profile. The values shows the average expression of the cells, or just the expression, I should say, probably here. And here we have each rows shows a cell, and then here we have the top 5 genes for given clusters: cluster 5, cluster 0, cluster 7. So if you look at these 3 clusters which have also been clustered together, we see that the top 5 genes show to a large extent more or less similar expression profile. And we can also probably say the same thing for these genes for these last 3 clusters, 11, 4, and 6. The dot plot shows more or less the same picture, but instead of having the cells in row, we are representing the overall expression, the average expression for the fraction of the cells in each cluster. Similar to dot plot that I showed you in a clustering section. So more or less the same picture, but here we have the extent of the expression in terms of this the colors that we see. For example, in cluster 7, gene FTL tend to show the highest expression, and apparently almost all the cells are showing this expression, and so on. You can also use violin plot, which not only shows the median expression by color, but also shows the distribution. So for example, let's look at the FTL one again, FTL. So here we can see in these 3 clusters, 507, we see that these tend to have higher expression distribution compared to others. So these will help you in case you have the gene marker to specifically look into that and see whether it fits well or aligns with your hypothesis. Last but not least is the matrix heat map. It's more or less similar to dot plot, but here you have the representation of the of the expression in terms of average expression, but it doesn't show you which fraction of the cells are showing that. Again, FTL1 seems to have highest expression in these three clusters, 507. Now, let's talk about comparison between conditions, COVID samples and control. I realize I have a typo here, comparison. Okay, good. So we can do that per cluster. And the way that we do it is to select a given cluster. And in this case, we are selecting cluster 4, for example. So it's not perfect naming. Because I'm labeling as cluster 1, but that's okay. It's just a matter of naming, so you can go around and play and change it. But anyway, so we select cluster 4, and then we print how many cells we have, and then we do the rank test. And in this case, we are specifying Wilcoxon test, and then we visualize top 25 genes. Okay, there we go. We have 300 COVID cells versus 293. It's more or less an even set. And there we go. Here we have RPS4Y1 and this. Interesting. So I will come back to this later. We can also show the distribution of significant genes. Like this, that we have the expression profile or distribution for each of these genes that we showed above, but from that there are 10 genes here. And in addition to that, we have control versus rest. This is COVID versus rest, and this one is control versus rest. And here, for example, we see CD74 and HLA. So these are related to immune cells, which tend to show different expression profile compared to others. But I will come back to this in next slide. Okay, so it's also possible to show expression distribution of a selected genes in each cluster for both conditions. And the way that we can do that, so here I am selecting 2 only. So we say get the data frame of this comparison. The group will be COVID, and the label of the test is used here, which is Wilcoxon, and then we extract the name of the genes. How many? 2. This will be for the first set, and the second set is when we are comparing control versus the rest. And here we are also comparing or extracting 2 genes. Then we concatenate them as a list. And then we generate a new data frame. And from our data, we will have the genes, and we extract these 2 columns, the type and the Leiden 0.6 clustering information. And we are not going to use the raw values. And then this is the second data frame that we are creating in order to summarize the information that we have in order to show it in a plot. What is the plot? On x-axis we will have Leiden, on y-axis we will have the value summarization of the previous data, and the colors will be by the type, and the kind will be violin type, and the color will be variable. So if we want to see what is happening under the hood, I will do this. I realize that this Copilot has been switched off accidentally. Now we have 1 more hour without Copilot's help. So anyway, so this is the table. We have the cluster information, we have the type, the variable correspond to the gene name, and then we have the value, which is the expression. So look at this one. Interesting. For example, for RPS4Y, we do not see very interesting expression pattern. For CD69, we may see some differences here and there, but it is not very clear. And when I see the Z popping up, it shows that we have a strong influence of the sex bias or imbalanced sex ratio in the data set. And CD74 seem to show some different expression profile, but I would say we should first control for the sex. And the way that we do it is that we remove the sex chromosome genes. So if you recall, we had tested it by biomarker to extract the annotation information, and here we are intersecting the gene names that we have in our AND-data object with the with what we have annotation information that we have extracted. We get the chromosome Y and chromosome X gene names, and then the sex, we make a union of the chromosome Y and chromosome X genes, and then we can see how many genes we have, and at the end we run it in a loop in order to generate indices that we would like to keep. And at the end we keep those genes which are not listed in chromosome X and Y, and then we rerun the clustering or comparison in this selected cluster. Okay, so just look at the numbers. We have 550 genes as sex genes, and then from 19,468, when we subtract, we will end up with 18,000 genes. And then when we do the comparison, we can see that the yeast and other genes coming from sex chromosome have been excluded. One thing which is important to note when it comes to the differential expression analysis is to see how consistent is the expression profile in the significant genes. And here we can do that by, before doing that, just for fun, let's see how will be these selected genes. The first two genes will look like after correcting for the sex genes. Okay, so there we go. Okay, and then we need to visualize it. I'm just jumping around. There we go. I should have copied all this. Okay, so here we have these genes now. If you look at the genes, so we do not see this, which we discussed. It was a specific X chromosome specific gene expressed in females, and now we have CD69, NFKBIA, CD74. So CD69, for example, and NKFBIA are slightly elevated in the COVID. This is pointing to immune activation and NFK beta pathway involvement. And at the same time, what we see is that CD74 is lower in COVID samples. We can specify a given clusters, but as a whole, the role of this gene is important because it is suggesting that there is a reduced APC activity. And last but not least, we have RPS26, which is largely stable with a slight trend towards reduced expression in COVID. So that's just a basic and very quick overview and interpretation of what we get from these plots. Again, we do see some biases when it comes to control. For example, cluster 11 is dominantly expressed in for NFKBIA is dominantly expressed in controls. Again, if you remember, we could see that this might be a bit biased since we have only controls in this cluster, and there are very few number of cells. So be careful when we are doing this type of interpretation. Okay, now let's go back to the checking consistent pattern of expression in significant genes. So here we are selecting top 5 genes. It's the same procedure, and here we are now plotting these two sets. The first one is COVID versus rest, the second one is control versus rest. And the violin plot looks like this. So there are some differences here and there. let's see if I can do rotation 90, rotation 90. I think this will look better. Yeah, it will be easier to read out. So for example, you can see that in some of the genes expression, we can see the expression profile is not consistent. For example, in gene RPS26, we see that there are 2 out of 4 controls which have lower expression distribution compared to the other 2. So that's important to check the consistency of this expression profile. Now we can also show that with dot plot. You have seen that before. And one thing which will be worthwhile to mention here in this plot and also in previous plots, for example, COVID-17, here and in this gene as well, we can see that has a distinct expression profile and has the, if you look at the count, it has the highest number of cells compared to the others. So this is very key piece of information when you are going to do the differential expression analysis, because if you are comparing unbalanced sets, some of the signals may be derived because of these unbalanced number of cells that you have. So in order to control for that, we specify a value, so then in the expression comparison, the groupings have more or less the same number of cells. The way that we do it is like this. We say, okay, the target cells to be 37, so it's like the maximum. If you look at the cell counts, I think it's not an off number, but we can play around with it. As long as it's more or less close to a balanced number, we should be fine. So that's how we first split the data by sample, and then we loop through, and then we say, okay, from each of these generated split samples, we want to have maximum of these target cells. And then we do the subsampling, and then we concatenate, and then we can check what are the samples sizes that we have. So we downsample to 37 cells per sample. Okay, so now we repeat the comparisons and visualize differential expression genes, the top 20 ones with the dot plot, and then we can see how it looks like. First of all, this is more or less the same, but not necessarily, I should say, because we do see some differences in the top, but I was biased because I immediately saw CD74. So I would say the expression looks better because we do not have such a huge variation among the samples. There are certain genes that still pop up, for example, CD74, CD69, which we have identified earlier, but this shows that they are a robust outcome. So it brings me almost to the end of this part. Please note that when you're examining data per patient, we can see some genes predominantly influenced by a single patient. It is generally a good practice to control for the number of cells from each sample when performing differential expression analysis. And here, that's how we did it. But there are also other several methods that can address these patient batch effects. They are mostly implemented in R, but it's possible to convert them to Python using RPY2. And here you can also use this link in order to see how it's performed. So currently we have not included this approach, but instead you can refer to patient batch effects section in the Sora differential gene expression tutorial from NBIS course, which I leave the link in the description. As a bonus, if you want, you can also do comparison between two selected clusters. For example, you may have two cell populations and you're interested just to characterize the differential expression between these two clusters. You use the same function, and here, when it comes to groupings, you specify the label of the cluster. The rest is the same. Here we have specified the correction method for multiple testing, and that's how we do it. And then we do some plotting, the matrix plot and the violin plot. So this will be smaller because we are looking into cluster 1 and cluster 2. That's the matrix map, and this is the violin plot. So you can see, for example, there are two genes, CCL5, for example, which is highly expressed with a distinct expression distribution in cluster 9 compared to cluster 2. Yeah. So I hope the overview that we had so far helps you to dip your toes into the ocean of single-cell RNA-seq data analysis, and the next step for you will be to challenge yourself by exploring larger data sets and more complex experimental designs. And remember, practice makes perfect. Good luck.